So when we talk about genic interaction, it is single character, and the interaction of more than one pair of gene is called gene interaction or interaction of genes. So these were the two scientists, Bateson and Punnett, who formulated this hypothesis of genic interaction. And they said this genic interaction is of two types. They are the non-allelic genic interaction and allelic gene interaction. Now, what do you understand by the term non-allelic? Located on different locus of the same chromosome or a different chromosome is called as non-allelic. You understand? You know what is a definition of allele? You know what is the definition of loci, right? Allele is a pair of gene which is responsible for a specific character. So this can be present in the same loci, in the same chromosome, or in different chromosomes also. So non-allelic genic interaction is the interaction between the alleles of different loci within the same chromosome or a different chromosome. What is allelic gene interaction? Interaction occurring between alleles of a single loci alone. Within the loci alone, there will be allelic interaction. So we will go one by one. So under non-allelic genic interaction, within your syllabus, we are going to study four types. One is complementary, supplementary gene interaction, inhibitory gene interaction, and lethal. Now, what is complementary gene interaction? When we study about what is complementary gene interaction, it is defined as two or more allelic dominant genes. I repeat the word, two or more non-allelic dominant genes it with each other to produce a character. But one gene cannot produce the character in the absence of other. It is like complementing. The word itself will give you the understanding. That is, these genes complement each other. They both will represent a character only when they both are present. In the absence of one gene, that character will not be expressed. That is what is called as complementary gene. Do you all understand, ma?
Yes, ma'am.
Asma? Asma, can you tell me what is complementary gene? Asma? Aishwarya? Aishwarya, can you hear me? Farheen?
Ma'am,
Farheen, what is complementary gene?
two or more non-allelic dominant genes interact with one another to produce a character, ma'am.
And
But
listen
one
completely just.
produce
Hmm.
that character in the absence of the other. If one is absent,
So,
the other can't produce.
okay, okay. So this is what is called as complementary gene. Then second one is supplementary gene. What is supplementary gene? It can be defined as two independent pair of dominant genes. Here it is two non-allelic. Here it is two independent pair of dominant genes which interact in such a way that each dominant gene produces its effect, whether the other one is present or not, because both are dominant, right? They both are dominant. They are not dependent on each other to represent their characteristics. So the two independent pairs of dominant genes which interact in such a way that each dominant gene produces its effect whether the other one is present or not, but the second dominant gene is added to the first to produce a new character. You remember the dihybrid cross?
Yes, ma'am.
Same way, yeah. So here there are two main dominant genes. Okay, so not necessary that they're dependent on each other, but if another dominant gene is present, a new character will be produced. That is what is called as supplementary gene. Okay, then third one is inhibitory gene. For this, we have another name called as epistasis. So here it is the prevention of the expression of one gene by another non-allelic gene. That is, the word itself, inhibition, gives you the meaning saying that one gene will suppress the expression of another gene. So epistasis mean itself stopping or inhibiting. So the inhibiting gene is called as the epistatic gene, and the gene which gets inhibited is called as hypostatic gene. You understand? The one which suppresses is called as epistatic, and the one which gets suppressed is called as hypostatic. So epistasis is of two types, namely the dominant epistasis and recessive epistasis. And then finally, we come to what is called as lethal gene. A lethal gene itself kills the possessor. For example, a plant or a human being who has the gene will not survive for these genes to reproduce for the next generation. So, lethal gene kills the possessor. Thus, the victim of lethal gene does not live to reproduce. So, these are the four main types of non-allelic interactions which you'll be studying for this chapter. We shall go one by one in detail. Now we shall start with the complementary gene. I told you, right? The complementary gene is, it is defined as two or more allelic dominant genes interact with each other to produce a character, but one gene cannot produce the character in the absence of other. For this, we have an example. This is the example. For example, you have two types of flowers. One is the white flower, which has the gene CCPP. Another white flower which has the gene CCPP, right? They both are hybrid plants, but the resulting F1 generation has a purple flower, CCPP, right? So when you draw this Punnett square, that is the checkerboard, what happens is you get a ratio of 9:7. You get 9 purple flowers and 7, what do you call as, white flowers. Okay, now how does this ratio come? See, this is one example which I took it from the net, but what is your textbook example? I will teach you in detail according to that. Now, see, are all of you attentive in the class now, please? So, the parents, we go to the parents now. When we talk about the parents, you have a white plant, a white flowering plant, and another white flowering plant, but the genotype of both these flowering plant is not a homozygous but heterozygous combination, CCAA CCAA. Now what happens is, C is a factor called as chromogen, whereas A is an enzyme called anthocyanin. Okay, so if a flower has to be red in color, this chromogen has to be activated in the presence of an enzyme called anthocyanin. So both the dominant genes should be present for the flower to give a red color. Do you understand, ma? So the chromogen is the factor which gives the red color, but it will give the red color only in the presence of an enzyme called anthocyanin. So they both are dominant genes and they complement each other because they have to be present in both. That is, the capital C and A both has to be present. Whereas here you can see in this white plant, this parent, you have the chromogen gene. But the anthocyanin gene is in recessive form, small a. That's why this plant is white, the flower is white in color. And in this parent also, you have small c. The small c is a chromogen which is recessive. But you have the enzyme, the anthocyanin, but that is dominant. But it will not represent itself. But in the next level, that is the FN generation, you take CA from this parent and CA from this parent. And when you get from both these gametes, you get the F1 generation, which has capital C, capital A, small a. So it has the dominant character chromogen and the dominant character anthocyanin. So ultimately, this plant will be red, flower will be red in color. Am I clear to all of you?
Yes,
Any
ma'am.
doubts?
Yes, ma'am.
Yes.
Mm.
No, ma'am.
So now what we do is we take this as F1 generation and go for the next part, which is the F2 generation. So for this F2 generation, we need four genes. That is gametes. So here, capital C, capital A, small capital C, small c, capital A, small c, capital A. So this is the sperms and this is the ova. This is how we take them as gametes. So in next slide, you can see we have taken the gametes here, right? You have the sperms here and the ovas here. So when you made them, for example, you take the one combination from here, one combination. So you have capital C, capital C, capital A, capital A. It is a red color flower. So again, this one, the same way as you did the dihybrid cross. This and this and this. So second line, the sperm from here and this ova. This sperm, this ova, this sperm, this ova, this sperm, this ova, spermatica. Okay. Third row, same way. One, two, three, four. One, two, three, and then four. So when you do this, obviously what happens is you get a Punnett square. Now what happens here is, one second. Okay. So you have got your combination. So when you see this combination, when you see this combination, you see you have a red flower, a red flower, a red flower, a red flower, because you have the capital C, capital C, capital C, A in both. A capital C, a capital A, capital C, a capital A, capital C, a capital A, capital C, and capital A. Same way here also. Capital C, capital A, capital C, capital A, capital C, capital A. And this way you see CACAC. Again a triangle just like what we did in the dihybrid cross. So all these triangles represent 9 number. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 you have red flowers. Now what about the white flowers? See, you have the dominant gene for chromogen, but the dominant gene for anthocyanin, which will allow this chromogen factor to bring that red color, is absent. You do not have a white, anthocyanin here. So obviously it's a white plant. No anthocyanin, white flower. No anthocyanin, white flower. Here you have anthocyanin, but no chromogen. You have anthocyanin, no chromogen. You have anthocyanin, but no chromogen. So this is also white. And finally, no chromogen, no anthocyanin, so white flower. So this is the combination, or what do you call the ratio of 9 is to 7. So this is the ratio of complementary genes. Do you understand what is complementary gene?
Yes, ma'am.
Are all of you clear with this?
Yes,
See,
ma'am.
one is dihybrid cross, monohybrid cross. That is the basics. The other is how the genes interact with each other. So you have the complementary gene, you have the supplementary gene, you have the lethal gene, and inhibitory gene. So today's class we have done with the complementary gene. Now what I want you people to do is, all of you, how many of you have done your whatever we have done in this class now in your notebook? Can anybody show? Can anybody show, ma?